in lipids, we call them, and other components. So what are those other components? The helper phospholipids that are also added include things like a molecule called diallyl phosphatidylethanolamine, which is a natural product and which causes cell membranes to fuse. So it's a fusogenic helper phospholipid. Cholesterol is also added to these formulations. Because it makes it so that the lipids around the RNA are not as rigid as they might be otherwise. There's the RNA, that's that strand that's kind of in turquoise color. And then there's the last thing, the lipid polyethylene glycol. People often they get confused about the use of the polyethylene glycol. This is a molecule that's been used in liposomes for therapeutic purposes for many years. It can cause the liposome and the lipid complex to be very difficult to gobble up by phagocytic cells, cells that are professionals at clearing virus and other particles out of the blood. And by using polyethylene glycol and putting it onto the surface of the lipid particle, it can make it so that those lipid particles aren't readily taken up and cleared in the liver, for instance. That is the classic reason to use polyethylene glycol, or PEG. The terminology is used, pegylation. For liposomes is to escape by the Kupffer cells in the liver. That is not why it's being used here. These cationic lipid RNA complexes have a tendency to bind to each other in solution and form very large aggregates. And when they do that, those large aggregates are no longer effective for transferring the RNA into cells, and they may be more toxic. And so it's important to avoid the aggregation or flocculation of these cationic lipid complexes, and that